Hi guys, welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator, and we are back in Bounce Back Re-NEET series. All right. So it is already confirmed by my side that audio video is normal and we can go ahead with the session. Give me a thumbs up that you're all set. And guys, this is part 2 of botany, and as you can see here guys, this is a very important opportunity, very important information for you, that you have this great opportunity to join the Prime membership for just ₹1, where you can watch all the lectures as per your need. If there is a lecture that you want to watch, in detail, although we are already discussing in detail, and still you want to attend your previous lectures that you have already taken in these batches, and you want to take more tests, you want to take all India mock test series, then don't forget to join this Prime membership. All right, good evening all of you. Good evening. Quickly share the session with all your friends guys. This class was promised to me 3000 years ago. Okay, King. Yes, I made this promise 3000 years back, you know. All right. So guys, how many of you have attended plant physiology yesterday? Quickly let me know. And don't forget to like, subscribe and share. Yes guys, how many of you have yes, taken, you have attended the previous lecture of plant physiology? Good evening all. You did? Rajitha, me, how many of you? Quickly give me a thumbs up. Say me. How many of you have attended the plant physiology, and how many of you tried to attempt the questions guys? Because tomorrow at 10:30 p.m. I am going to take a special class on Unacademy Learners app. I have already scheduled this class for tomorrow guys. So don't forget to follow me on Unacademy Learners app and take these free question practice session, where we'll be discussing all different types of questions that are coming in NEET exam, statement based, match the following and all of that. So do that today. And I've already uploaded the notes, and I'm going to start with today's session guys. In today's session, we are going to discuss 3-4 chapters today. Number 1, plant kingdom. I'm going to start with a heavy topic. Okay, plant kingdom with the help of flowcharts. How many of you found those last class flowcharts useful? Tell me. How useful were those flowcharts? Yes, guys. How many of you found those flowcharts very useful? Quickly let me know. Good evening. Yes, Jack. Good evening, Nimish. How many of you found those flowcharts useful? Very useful. Sandhya says, wow. Cool. Very useful, right? Now today, the flowcharts that I have brought for you for this chapter of plant kingdom, biological classification, the mnemonics that we are going to discuss, and I'll try to finish anatomy also today. You know, there's a trick to study anatomy. So we are going to discuss it all in today's session. So give me a thumbs up in the beginning itself. Let's have a powerful energy in the beginning, and we are going to start with that. Quickly. Are you making the notes with me, or you are just using my notes? Yes, please. Are you making your own notes? Flowchart was promised to us. King! All right, guys. If you liked last class notes, then you are going to love these notes. Love them. Let's start with plant kingdom. So guys, yes, starting with plant kingdom, there are different types of classifications. First of all, we have artificial system of classification, which was given by Aristotle and Linnaeus. Now, this artificial system was based on some superficial criteria, so not very authentic. And then we have natural system given by Bentham and Hooker. You can just get such questions in the match the following type as well. So this method is more authentic because it is not based on just superficial criteria. It will include all the internal, external structures, phylogeny, everything. We'll go into the details. Then we have chemotaxonomy, in which the classification is based on the chemical properties. Then cytotaxonomy, which is based on the cell structure, the chromosome number, right? Then numerical taxonomy. This one taxonomy can consider so many criteria at one time. See, there are limited number of criteria considered in artificial. Then natural system considered more number of criteria as a basis of criteria for classification. Hence, it was more authentic. But numerical taxonomy is that system of taxonomy which is going to consider hundreds of characters simultaneously. Why? Because it is not just human, it is obviously not humanly possible. Numerical taxonomy is going to make use of computers. So everything will be fed into the database. All the characters will be fed there. So hundreds of characters can be considered, right? Every character is given a particular code number. So in numerical taxonomy, every character is getting equal weightage, right? And then we have phylogenetic system given by Angler and Prandtl. Now, I'll come back to this in a while when we discuss the biological classification. I want to come quickly to plant kingdom because right now the energy is high and I want to make the best use of that. Now see, in plant kingdom guys, we are going to see three types of life cycles. One is haplontic life cycle, another is haplodiplontic life cycle, and the third is diplontic life cycle. Is that clear? We will be understanding it one by one. Now, life cycles may not be there in your NCERT as a proper topic, but life cycle is in your syllabus. And since last year, guys, we are getting sequence-based questions, sequence of the life cycle-based questions. So be very, very attentive while we learn these, while we understand these. Is that clear? Now, first of all, let's understand what is haplontic life cycle. In haplontic, the word comes from haploid. Okay, so haplontic life cycle, just remember, in this, in most of the lifetime of the plant, in most of its lifetime, haploid phase will be present. Diploid phase, if of course it will be present, diploid phase when present will be very short-lived. See, I'll start from zero again. Remember, whatever the plant type be, whether the plant is haploid or diploid, whatever the plant be, both the phases have to occur in the life cycle of the plant. Plant will come across diploid phase, plant will have haploid phase. Now it depends on the dominion or the domination of a particular phase that we give them certain names. For example, most of the algae, I'll not say all the algae, please. Most of the algae will have haploid phase for most of the lifetime. Hence, we say most of the algae is haplontic. Is it clear? The main parent body is haploid and diploid phase is very, very short-lived. We'll see how and why and when and where, right? Then coming on to haplodiplontic. Now, in this, as the name says, there are two words, haplo and diplontic. So, the plant body will have sufficient haploid phase and sufficient diploid phase as well. So, the two will have their own prominent existences, right? So, and then we have diplontic life cycle, very opposite to haplontic. Like in haplontic, most of the life phase was haploid and diploid phase was very short-lived. Now, just opposite to that, in diplontic life cycle, the diploid phase will be super present and it will be dominant and it will be present for most of the time, while haploid phase will be very, very short-lived and reduced. Is this point clear? I hope the word meanings are clear. Please let me know that these points are super clear to you. Yes, guys? Ma'am, sir is taking class. Which class, guys? Yes, guys. So, I think I already... Physics class is going on. Yavar sir is taking class. How long is it going to go? Because I have already... I think I didn't know that. That's why I already delayed the class. Yes, yes, yes. Don't worry, guys. Don't worry. All right. Are these points clear? Are these points clear? Never mind. Those who are not able to attend the class live, they'll be able to watch it later on. But those of you who are present here, please concentrate. Is that clear? All right. Never mind. Never mind. All right. So guys, this is the time when we have to take so many things simultaneously, right? Never mind. All right. Now, so I hope this point is clear. So as a matter of fact, most of the algae will have haplontic life cycle, right? But some algae will show haplodiplontic also and diplontic too. But bryophytes and pteridophytes will have haplodiplontic life cycle, while gymnosperms and angiosperms will have what? Diplontic life cycle. Are these points clear? Are these points clear? No problem. Yes, guys. All right. Clear? Perfect. Now moving on. So I had a word with Yavar sir, no worries. Let's continue. Now we will start with first of all, that is algae. Let's start with algae here. Now see, in every life cycle, there will be the dominant plant body which is known as parent. Now learn few things very clearly. How are parents? Be it plants, be it algae, be it bryophytes, be it pteridophytes or gymnosperms or angiosperms, the dominant plant body is known as parent. And what are the typical characters of parents? Be it parent of the plant or parent or parents. Parents, being parents, have some typical characteristics. How? See, parents are always, see, wait. Yeah. Now see, whatever be the type of the plant, be it, as I said, algae, pteridophyte, gymnosperms, whatever, parent will have certain characteristics. Okay, they will always be independent, they will always be dominating, they will always be free living, they are always autotrophic. Is that clear? They are dominating. I hope these points are clear to you. These points are clear. All right. Now see, when we are going to go into the details, let's first understand the evolution of plants also. See, plants can be divided into two broad categories. One without seeds and the other with seeds. So there are seedless plants and then there are seed bearing plants. Is that clear? Now seedless plants are also flowerless. We call these seedless plants as cryptogams, while the seed bearing plants will be known as phanerogams, or we call them spermatophyta. Here the sperm word is referring to seeds. Is that clear? Now, first let's go into the details of cryptogams, that is seedless plants. Now the plants which are seedless, they are further divided into two categories. Plants plant body which is not very well differentiated, that means they don't have a typical root, stem or a leaf, they don't have, right? So such plant body is known as thallus. And in these plant bodies, in this group of plants, the vascular tissue is also absent. Now this includes two different categories, algae and bryophyta. Is it clear? So algae is seedless plant, body is undifferentiated, that means no true root, stem and leaf, and they also don't have vascular tissues. And so this is algae. So we also call algae as a thallophyta because the body is thalloid. But coming on to bryophytes, their body is also undifferentiated, but it has started developing some root-like structures, stem-like structures, leaf-like structures. Okay, so we'll not call it thallophyta because it is little more differentiated, but still true leaf, stem and roots have not developed, and the vascular tissue is absent in both. Is that clear? Then there is another type of seedless plants or cryptogams where the body is differentiated. So are you able to see the evolution here? Now the plant body has become differentiated. Vascular tissue will also be present, but seeds are not present. So again, under the category of cryptogams. Now this type of plant is known as pteridophyte. Now why I have drawn this flowchart? Because you do get questions like a botanist has identified a plant with such characteristics. Which of the following category will it belong to? So you should be able to identify that if it has these characters, what will be the category of the plant? I hope you will be able to answer such questions now. Right? Then we have seed bearing plants, that is spermatophyta or phanerogams, and here vascular tissue has already evolved. Now vascular tissue will be present here as well. Now seed tissue well differentiated. Now the seeds are present, but the seeds are naked. Now this comes under gymnosperms. Was this question there in 2026? Yes or no? So the naked seeds are found in gymnosperms. Why? The seeds are not enclosed within the fruit. Clear? So they don't have flowers. They don't have flowers, and that is why the since they don't have flowers, the seeds are not enclosed within the fruit. Now here flowers will also be present. So seeds are enclosed within the fruit. So this includes the plants coming in this category called angiosperms, which are further differentiated into dicots and monocots. Please tell me that these points are clear. Please tell me these points are clear. Yes or no? All right. Now, listen. So this is another table to simplify things for you. See, plants on the basis of vascular tissue now. There are non-vascular plants and then there are vascular plants. The vascular plants will be known as tracheophytes. Now see here. In non-vascular plants, we have two categories, algae and bryophytes, and the body is not differentiated, so they have thallus-like body. Right? So in algae, see, vascular tissue absent, root absent, stem absent, leaf absent, seed absent and flowers absent. Then we have bryophytes, vascular tissue, root, stem, leaf, seeds all absent, flower is also absent. But instead of root, stem and leaf, these bryophytes will start developing some similar structures, root-like rhizoids, right? Leaf-like thallus. Is that clear? So similar structure will start developing here, but still the body is thallus-like and they are non-vascular plants. Then coming on to vascular, we have three categories here, pteridophytes, gymnosperms and angiosperms. Now in pteridophytes, vascular tissue is also present, root, stem, leaf have also developed, seeds are not formed, flowers are not formed. Here in gymnosperms, all the things are present, but flowers are not present, so seeds are naked. And in angiosperms, again, all things are present, flowers are present, so our seeds are enclosed within the ovary. Do you see this table? Is it helpful? Please tell me, all these points are clear? All these points are clear? Yes or no? Shall we move now on? So, all right. So, talking about algae now. Now see, algae. Algae also known as thallophyta. No true root, stem, leaf, no vascular tissue, no seeds, no flowers. What are the other features? So, the body is simple. Body is thalloid. Now, it is a plant, so it is photosynthetic, hence it is autotrophic. Now, see, there are certain algae which are going to show some symbiotic association as well. See, algae and fungi will form lichens. Algae also shows symbiotic association with an animal known as sloth bear, who is so lazy that it doesn't move at all to get its food. So there is algae growing in its body, in its fur. So it takes out algae from its fur and eats it. It is so lazy that sometimes it even dies of starvation. Now, other features. Now, algae is mostly aquatic. It is either found in fresh water or marine water. It is mostly aquatic, therefore it does not need any vascular tissue, or it is found in the moist soil, stones and wood. Are these points clear? Now the size is very variable. It is microscopic also, it is massive also, and it can be unicellular or multicellular. Now see, Microscopic ones are Volvox and Gilothrix. Massive ones which are 100 meters tall are kelps, and unicellular ones are Chlamydomonas. Now see, there are other examples. They live in different forms. I told you they can form colonies. Again, Volvox, they can be filamentous, thread-like structures. Eulothrix and Spirogyra, and they will have massive structures 100 meters tall. It is huge. So it is found under oceans only, and it is known as kelp. It is known as kelp. Is that clear? Right? Is that clear to all? Now, moving on to the next. So, reproduction in algae. See, so reproduction is of three types that is seen in algae. One is asexual, vegetative, and then sexual reproduction. See, vegetative method is very easy. You cut the fragment of Eulothrix in different parts, and every fragment will develop a new plant body, right? Eulothrix, Spirogyra. So all those which are filamentous will show fragmentation, right? Then talking about asexual method. Now in asexual, there are going to be some spores formed. Now spores will be formed here as well, but spores will be a part of sexual reproduction as well, but then the main thing is gametes here. So what are the different types of asexual spores found in algae? So the most common ones are zoospores and eplanospores. Now see, zoo word is for animal, animal is motile. So remember, the motile spores are called zoospores and they are flagellated, while the non-motile spores, that means they will not have flagella, they are known as eplanospores. Now see, these terms can be used at multiple places. They are generic terms. Usually every motile spore is known as a zoospore. Is that clear? We might have specific terms, and when we don't get a specific term, we call a motile spore as zoospore, and zoospore is always asexual. Is that clear? Now coming on to sexual reproduction. So gamete formation is a characteristic feature of sexual reproduction. So gametes are of three types. Now see, in certain plants, in certain algae, male and female gamete is just similar. It is very difficult to differentiate or identify which one is male or female. When there is similar male and female gametes, this condition is known as isogamous. This condition is known as isogamous. Then, if male-female gametes are dissimilar, we call it anisogamous. And then there is a third type called oogamous, in which the male gamete is small and motile, while the female gamete is large and non-motile. Very important. Now again, male female are different. So basically oogamous oogamous condition is one type of anisogamous, but we keep it separately. Please tell me these points are clear. Is this clear to all? Is this clear? Now we'll have to see that which algae show isogamous condition, which are anisogamous, and which are oogamous as well. Now talking about the economic importance, this flow chart is so important. We get many questions from this flow chart. See, don't just keep NEET 2026 as an agenda. There have been exams before that and good exams before that. So consider those questions also. Okay. Now see, economic importance. First of all, algae is the maximum oxygen producer in the aquatic habitat, right? More than half of the carbon dioxide fixation, that means photosynthesis, is done by algae. Second, it is a main producer, as I told you, for the aquatic food chain. In aquatic food chain, it generally begins with, see, every food chain, the grazing food chain will begin with the producer. So who are the main producers in aquatic habitat? It is algae. Then there are certain algae which have water holding capacity. Is that clear? They produce some water holding substances like algin and carrageen. Such algae, such water holding substances are known as hydrocolloids. Is that clear? Now why do we use them? Water holding substances will be used for transport of seedlings and plants. When we have to transport them for across the long distances, we are obviously not going to take them in pots and keep watering them every now and then. No. We pack them in these hydrocolloids. They will absorb water, they will retain water, and they'll stay moist for very long time. Is that clear? So remember, algin and carrageen, these are important PYQs. So algin is obtained from brown algae and carrageen is obtained from red algae, ABCR. This is how I remember. Clear? Then there is this agar. This agar is obtained from Gracilaria and Gelidium, which is red algae, right? Now, agar is used in ice creams, jellies, and also agar is used as a culture medium and used in cosmetics. Then there are certain algae which become a food source also, which is eaten. For example, sargassum, laminaria, chlorella, spirulina. You must know that chlorella and spirulina are used as space food. They are rich in proteins and they are called single cell proteins, right? And then laminaria is iodine rich. It is rich in iodine. Please tell me these points are clear to you all. Is that clear to you all? Right? Now, moving on. All right. Just a second. Please tell me these points are clear. Quick. Is that clear? Now, we have three classes of algae based on their pigments majorly. Now remember, we have learnt it yesterday that all those organisms which have chlorophyll and are photosynthetic, they will always have chlorophyll A. So chlorophyll A is a universal pigment. Have we read it yesterday? Yes, in photosynthesis. Now we are going to see this part here. See, so based on pigments, with the green pigment being dominant, we have chlorophyce, the green algae, we have phaeophyce, the brown algae, and we have rhodophyce, the red algae. RR, remember? Okay. So, in chlorophyce, in all of these, chlorophyll A will be present. So, just remember it in a pattern. So, I remember it CPR. See here. So, the pattern is supposed to be remembered like CPR. Chlorophyce, phaeophyce, and rhodophyce, right? All of them will have A, and then remember the pattern B, C, D. Can you learn it like this so that there is no confusion? Remember CPR and then go in a pattern. A is a universal, all will have, then B, C, D, right? Now, other than these, phaeophyce will have additional pigment known as fucoxanthin, which is actually a xanthophyll. And other than this fucoxanthin, it will also have carotenoids, right? Then talking about rhodophyce, it has this red pigment known as phycoerythrin. The name itself says erythrin, so it will be red in color. And then this only has A and B. Are these points clear? Now see the examples, how to learn them. So told you chlorophyce is green algae, and what are the color variations? Bright green to dark green, brown algae olive green to brown, and red algae red in color, actually red in color, right? These are different examples. Volvox, Chlamydomonas, Eulothrix, Pyrogyra, Cara. We don't have to, Udogonium, yes, learn that, and Cladophora. Okay, so Cladophora was that algae which was used in that prism experiment. Can you tell me who was the scientist who did that prism experiment, which we have read about in plant physiology, in photosynthesis? Yes, guys, let me know. Who used that prism experiment quickly? Yes. Guys, please stop wasting your time. Engelmann? Are you sure it was Engelmann? Yes. All right, perfect. So it is that Cladophora. And then we have phaeophyce examples: Kelps, Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. See, this mnemonic was made by one of my students only. So ecto helps Sargun learn dicot function. So ecto is Ectocarpus, helps, it rhymes with Kelps. Sargun is Sargassum, learn is Laminaria, dicot is Dictyota, and function, from function we remember the Fucus. Are these points clear? And then for this, we have P2G2, Porphyra, Polysiphonia, Gelidium, and Gracilaria, and we have just read these two are the sources of agar. I hope these points are clear to everyone, right? We have talked about their pigments. Now talking about their stored form of food. So that table given in your NCERT is very, very important. We have seen multiple PYQs coming from that table. So I have elaborated that table and added more points to it. So stored form of food is starch plus oil droplets. Now in case of Chlamydomonas, guys, there is a special structure which is known as pyrenoid, Now this pyrenoid is a storage body which contains starch and protein. Very important to remember. What does pyrenoid contain? It contains starch plus protein. So this is a Chlamydomonas I have drawn, and it has a typical cup shaped chloroplast, right? Now see, in Phaeophyce, the stored form of food is mannitol or laminarian starch. And in red algae, this is Floridian starch. And the structure of Floridian starch is, guys, very similar to amylopectin and glycogen. We have already discussed glycogen in biomolecules. Right? Now talking about the structure of cell wall, all of them will have cellulose being plants. Cellulose is a typical component. Cellulose is present in all, but in Phaeophyce, elgin will also be present. While in Rhodophyce, apart from cellulose, we will have pectin and polysulfate esters. Please tell me these points are clear. Now, if I talk about the number of the flagella, so there are 2 to 8 flagella in case of Chlorophyce. Okay, and the 2 flagella are equal and on the tip of the cell. So 2 and apical. But in Phaeophyce, again, the 2 flagella are present, but they are not equal, they are unequal. Do you see? And they are not at the tip, they are lateral. So do you see there are 2 flagella arising from the side, right? And one of them is short, the other one is long. Now see, the short one shows a whiplash movement, while the long one is tinsel shaped. That means it has hair-like extensions which increases the movement. So the main flagella which is going to generate and decide the direction of movement is the short one, right? And then we talk about the Rhodophyce, flagella are absent, and we will see the absence of flagella even in its gametes. Are these points clear? Yes or no? Right? Then talking about thallus, the thallus here can be unicellular, multicellular, it can be colonial or filamentous. While here the thallus is multicellular and it is differentiated into 3 regions. So it will have a typical, it will try to resemble a leaf-like structure. So we have this holdfast, we have this stem-like structure called stipe, and it will have leaf-like structure known as frond. So this is frond, this is stipe, this is holdfast. Are these points clear? So holdfast will act like a root and it is meant for attachment for the substrate. It will not absorb anything. Then stipes are stem-like structures, while frond is leaf-like. It has midrib and it will contain these air cavities. Okay, air bladders, and these air bladders will keep it floating. It will provide buoyancy. And of course, this frond is photosynthetic. Is that clear? And then Rhodophyce will be multicellular and it will have a very complex, complicated thallus as compared to rest of the 2. Talking about the habitat, mostly freshwater, few brackish and saltwater, mostly marine. Now in NCERT it is written rarely freshwater, and for Rhodophyce it is using the term few freshwater. Hardly any difference, but in order to remember, now in order to remember this, see, NCERT is very direct sometimes, and it doesn't accept any, sometimes it happens that it just goes lakir ka fakir. Do you understand that? Very direct, no deviation at all. So whenever it has written rarely, think of Phaeophyce. When it is writing few, think of Rhodophyce. Is that clear? In case there are no other characters given, then this can be your point of distinction. Otherwise, rest is fine. Are these points clear? Please tell me these points are clear to all. Yes, guys, please tell me these. Are these points clear here? Shall we move on now? Right? So guys, we are talking about algae. Now talking about different types of reproduction in all the different types. See, Chlorophyce, Phaeophyce, Rhodophyce, fragmentation will be seen in all of them. Fragmentation is seen in all of them. Is that clear? Talking about asexual reproduction. Now see, in chlorophyce, there is formation of zoospores, and zoospores are always flagellated and motile, and the structure where they are formed is called zoosporangium. Now in phaeophyce also zoospores are formed, but do you see the difference between the zoospores? They are both motile, they have flagella, but here the flagella are two and apical and equal. Here the flagella are two, lateral and unequal, right? This is what we read about flagella. While here they will form non-motile spores, which will be known as aplanospores. We have read that rhodophyceae don't have flagella, so they will not be present in their spores or even in their gametes. Now talking about the sexual reproduction. So the gametes will be formed in all. Now in chlorophyceae, gametes will show all the three conditions. Iso and oogamous. Here also it will form iso and oogamous condition, while in sexual it will form only, in case of rhodophyceae, it will only show oogamous condition. Are these points clear? Please tell me. Are these points clear? Yes guys, quickly let me know. And now I am going to discuss the life cycle of algae. Now algae, as I said, will show all the three different types of life cycles, but it is mostly haplontic. So I am going to discuss the haplontic life cycle. Now see, as I said, in haplontic life cycle, the parent is N. Haplontic means most of the life phase will be haploid, right? So the parent here is going to be N. So 2N phase is going to be very, very short-lived and reduced, right? So who is going to represent the 2N phase? We'll see it. All right, guys. So talking about the parent, so parent is haploid. Now remember certain terms. Haploid phase or haploid body will be known as gametophyte. Is that clear? Can you remember certain things? Yes, please. Can you remember? Gametophytic phase is the term used for haploid phase, right? Also, as the name says, gametophyte. Gametophyte is the one which is going to produce gametes. First, just learn these things by heart. Even if till now you have been getting confused in these terms, I want you to just be very thorough with these terms now. Incubate over it. Sit on these terms, think of them again and again, learn them by heart. Whatever might go wrong, but you should never go wrong with these terms. Is that clear? Now, second is sporophyte. Sporophytic phase is the diploid phase. So sporophyte, as the name says, is the one which produces. So sporophyte is going to produce spores. So the name itself says what it is going to do. Gametophyte will produce gametes. Sporophyte will produce spores. Yes. Also, gametes are always haploid. So gametophytic phase will be haploid. Sporophytic phase will be 2N. Are these points clear? Please give me a thumbs up that these terms are super clear here. Yes, guys. Are these terms clear to you? Yes, please. Let me know. Are these terms clear to you? Yes? All right. Now, let's come back to this life cycle once again of algae. Now, see. Parent is haploid. So, what does the parent body represent? Parent body represents a gametophyte. And I've just told you what is gametophyte, which produces gametes. So gametes are always produced in the sex organs. Now, things are linked. So this is parent. Parent is haploid. Parent haploid, and therefore it represents a gametophyte. Because it is a gametophyte, it is going to form gametes, and gametes are formed in the sex organs, right? So parent will have sex organs. Is that clear? So there will be male sex organ, there will be female sex organ. Now the sex organs will also be haploid. Why? Because the body itself is haploid. So sex organs are haploid. Are these points clear? Okay. Jao. So now see what happens. This is the male sex organ and this is the female sex organ. They are already haploid. Gametes are haploid. So if something has to form, if you have to form haploid from haploid, what type of cell division will occur? Mitosis. So mitosis occurs in these sex organs, and that is how gametes are formed. So you have to be very clear how and which cell division is taking place. Remember, if you want to maintain the same ploidy, then it is mitosis. If you want to change the ploidy to half, then it has to be meiosis. It is already haploid. Gametes have to be haploid for sure. So haploid to haploid, it will be mitosis. Is that clear? So gametes are formed by mitosis. Now gametes are going to undergo fusion, that is called fertilization, and it is a rule whenever the gametes will fuse, zygote will be formed. Zygote will be formed, and zygote is always 2n. Zygote is always 2n. Now see, zygote will quickly undergo zygotic meiosis, right? Zygote will not multiply to form a multicellular structure. No, zygote, this one cell is immediately going to undergo meiosis to form spores, going to form spores. Please tell me these points are clear. Now because this is 2n and this is undergoing meiosis, so spores formed will be haploid, and now haploid spores will germinate in favorable conditions to form the gametophytic body again, right? Now from n to n, you have to form n to n, then what type of division is going to occur here? It will be mitosis. Also, germination always involves mitosis. No matter where germination occurs, it always involves mitosis. Are these points clear to everyone? Please tell me. Please tell me. Now, I have already written what are the other features of the parent. I told you, be it our parents, be it the parents of algae, parents are always same. They are always dominant. They are always autotrophic. They are always independent. They are always free living, right? And here we have to remember they are multicellular. Are these points clear? Who is called a parent? The dominant plant body is called a parent. Are we clear? Please tell me. Now see, I have written this. I've made this half triangle. Now see, the most of the phase is N, right? So that is why we call such life cycle as haplontic. Now do you see there is a very short-lived 2n phase coming here, which is represented by zygote. Learn these lines. 2n, that is the diploid phase or sporophytic phase, is represented by zygote. It is short-lived. It quickly undergoes meiosis to form spores. Remember these lines. So sporophytic phase is very, very reduced here, while majority life cycle represents the gametophytic phase. Are we clear? Now, can you answer these questions that the sporophytic phase in algae, haplontic algae, is represented by? Can you answer these questions? Please tell me, give me a thumbs up that you're able to understand everything. Fucus is diploid. Yes, fucus is diploid. Fucus is going to show diplontic life cycle. Volvox will also show diplontic life cycle. I am talking about the majority of the algae which is haplontic. Is that clear? Now, so we have seen this. So already told you, see, majorly haplontic. Some of them will show haplodiplontic and others will show diplontic phase. So you have to learn which ones are haplodiplontic, which ones are diplontic. Can you answer that? Yes, who can answer this question? Name the algae which is haplodiplontic. Quickly. Name the algae which is haplodiplontic, guys. Quick. And those which are diplontic. And those which are diplontic. Quickly, let me know the answer. And now, thereafter, we move on to the next phase of plants, which is bryophytes. We've discussed this all about algae. Clear? Now coming on to bryophytes. Now see, algae is found within water. That means it is mostly aquatic. Now from water, it will start coming out on land, but it will not quickly and suddenly jump onto land. It will show gradual movement. So now algae was completely in water. Now we have bryophytes. They are near water. They will have their life cycle partially in water and partially terrestrial. So such plants are known as amphibians of plant kingdoms, that is your bryophytes. Because why amphibians? Because it needs water to complete its life cycle for the sexual reproduction. Also, so it is found in moist shaded areas or damp humid shady areas. Now see, this is the first line of your paragraph, NCERT, and right here we get two assertion reason questions. They can give bryophytes are called amphibians of the plant kingdom and it needs water to complete the life cycle for sexual reproduction. This is assertion, this becomes a reason. Or they can say bryophytes are found in the moist shaded areas, damp humid shaded areas, and it needs water. So this becomes the reason again. So whatever be the assertion, the reason remains the same. Please tell me these points are clear. Please tell me these points are clear. Quick, quick, quick. Right? All right. Now, coming on to the features of the bryophytes. See, they're called amphibians. We have already seen algae and bryophytes. The body in both these plant categories is not differentiated. Algae is not at all differentiated, but we'll see some basic differentiation in bryophytes, but still it does not have two leaf, stem and root. Okay, so body is still thalloid but not differentiated into two root, but it is certainly more differentiated than algae. So it has root like structure, right? It has root like structure known as rhizoids, but unlike roots, roots will help in attachment as well as in absorption, but unlike roots, rhizoids are only meant for attachment. They will not be involved in absorption. Is that clear? Secondly, rhizoids are always branched. The roots may or may not be branched. Root hairs may or may not, root hairs are not branched, right? But these rhizoids can be, they will be branched. Root hairs are always unicellular. We are going to discuss that in anatomy, but rhizoids can be unicellular as well as multicellular and no role in absorption at all. Are we clear? It does have, bryophytes will have stem-like structure. They will have leaf-like structure as well. Please tell me these points are clear. Yes? All right. Moving on. Now, algae, the main plant body was gametophyte, but in bryophytes, we are going to have haplodiplontic life cycle, one step ahead. But the main plant body or the dominant phase or the parent, main plant body is known as the parent. So parent is going to be haploid. That means a gametophyte, and life cycle will be haplodiplontic, haploid phase being the dominant phase. Are these points clear? Now, it will have the diploid phase also, which will be known as sporophyte. But in here, sporophyte will be prominent, but it will be attached to the gametophyte. We'll see the structure in detail. It will not have independent existence and it will be differentiated into three regions: foot, setae and capsule. Since you have already read about it, I have drawn this flowchart where you can understand these things. Is that clear? Now, this economic importance of bryophytes is also very, very important. One, economic importance of algae, and second, that of bryophytes. Both are important. Now, first thing first, bryophytes are known to act as pioneer species in xerophia and play important role in succession of bare rocks. Now, understand. What are pioneer species? Pioneers are those who lay the foundation. You must know that wherever you see a forest standing in present day, that area was not always that lush green. Every forest has a huge story and a long story of evolution. So it began with rocks. See, there's a difference between rock and soil. So rock is hard and nothing can grow on rock. So first, rock should weather, rock should melt down to form, or it should break down to form soil, right? Now, how is rock going to form soil? There are multiple factors which can convert rock into soil. One of the factors, living factors, is lichens and mosses. These are, lichens are not bryophytes, but mosses are bryophytes, right? But together, both lichens and mosses, they secrete some enzymes which are going to corrode the rock, which are going to cause weathering of the rock and form soil. And once the soil is formed, that's when the story of vegetation begins, from herbs to shrubs to small trees to tall trees, and then developing into a stable forest. So there's a long history of every forest, but it starts with lichens and mosses who are going to break down the rock into soil. So the first vegetation which inhabits any rock area, any bare rock area, is lichens and mosses. So being the first ones, we call them as pioneer species. Are these points clear? Xeros here means when the succession occurs on the rock area. Now, Second one is it acts as a food for herbaceous mammals and birds. Algae is also going to act as a food. You must be able to recall that which algae are eatable, which are edible, which become food source. Then these bryophytes, they are eaten by mammals and herbs, herbaceous mammals and birds. Then there is certain bryophyte which can be used as a fuel. For example, sphagnum, we call it a peat moss. Okay. Then some bryophytes, again, they act as a packaging material. Algae was acting as a packaging material also because it has water holding substances called hydrocolloids. Now here these bryophytes will also be used as packaging material for transshipment of living materials because of the water holding capacity again. So here also we get assertion reason. Then another function. Now mosses, they will form a mat over the soil. So because they form a dense mat, it is going to prevent the soil erosion. Are these points clear? Then coming on to the life cycle. Now see, this is more elaborate, right? Because now it is moving towards haplodiploidic things. Now see, first of all, the dominant body of bryophyte is also haploid, and the dominant body is known as a parent. How are the parents? Parents are dominant, autotrophic, independent, right, free living. These are the general characters. Then the parent body of bryophyte is also haploid, that is gametophytic, right? And we have read that bryophytes body is still undifferentiated, does not have true root, stem and leaf, but it has root-like rhizoids. We've seen the structure, some features of rhizoids. Then it has stem-like structure, root-like structure, right? So it has rhizoids, main axis and leafy structure, right? Now this is also a gametophyte. So can we recall? Gametophyte is going to produce gametes, right? And in order to produce gametes, it should have sex organs. Now see here, parents will bear sex organs. Now in case of algae, the sex organs were non-jacketed. Remember that. In case of algae, where is it? Yes, in case of algae, guys, the sex organs are non-jacketed. See, jacket is like a cover, right? When we are putting a jacket on, we are putting a cover on our body, right? So these sex organs are not covered, while in bryophytes, the sex organs will be jacketed. Now you will be able to understand what does a jacket look like. So jacket is nothing but a single layer of cells which surround the sex organ. See here. So this pink structure, this represents the female sex organ which is known as archegonium, and it is flask shaped structure, right? Do you see I have drawn a single layer of cells around it, right? This single layer of cells is going to be known as a jacket. So sex organs are jacketed. There are two parts. We call this part as neck and this basal part as vent, and within the vent we will have a female gamete sitting. Is that clear? So parents will have sex organ. Male organ is known as entheridium, female organ is known as archegonium. Again, the sex organs are haploid, right? Now female organ, sex organ is flask shaped and jacketed. Entheridium is going to produce male gametes, archegonium is going to produce the female gamete. Female gamete is also known as egg. Is that clear? While male gamete is known as entherozoids. Now see, entherozoids are biflagellated. These are what entherozoids look like. They have two flagella, and then after being produced, they are released in the water. Remember, bryophytes need water for completing their life cycle. So they will be released in the water. Now, female gamete or the egg is non-motile. See, the female egg will remain here. This entherozoid is released in water, and swimming in water, it will come to the mouth of the archegonia. Do you understand that? And it will come to the mouth of archegonia and go deeper into the vent, and this is where fertilization will take place. Please tell me these points are clear, right? Now, after fertilization, it's always the time for zygote formation, and zygote is always 2n, right? Now, unlike in algae, where zygote immediately resulted in meiosis, it underwent meiosis, that's not the case here. Here the zygote is going to undergo mitosis and form a multicellular structure which will be known as sporophyte, because since it is a derivative of zygote, it is 2n. So it will form a 2n structure, a sporophyte, a multicellular structure, but this sporocyte is not independent, it is not free living, it will be attached to the gametophytic body. Now let's say the gametophyte looks like this. Let's say gametophyte looks like this, the leafy shoot. So these are the rhizoids, this is the main axis, and it has leafy structures surrounding. So these are the leafy structures present in the parent body, right? This represents the sporophyte. Do we understand this? Rhizoids, main axis, and the leafy structures, not leaf. Now, the sporophyte formed will be attached here. This is where the sporophyte will be present, like this. It is attached to the parent body. Why? Because it has to draw nourishment. It is attached to the gametophyte for nutrition. Can we call it partially parasitic? Yes? Okay. So the three parts of sporophyte are foot, seta, and capsule. Now see, this top structure, this apical region, this is called capsule. Capsule will have some spore forming cells. So spore mother cells, right? Now these spore mother cells are again diploid because we are talking about a diploid phase. So these spore mother cells, where are they present? Within the capsule. Spore mother cells will undergo meiosis to form haploid spores. And with the formation of spores, the gametophytic phase will begin again. Spores will find the favorable condition to germinate and give rise to the new parent body. Please tell me these points are clear. Now you see, from the time of zygote formation till spores, sporophyte, having foot, seta, and capsule, this is all which phase going on? 2n phase going on, that is the sporophytic phase. So here the sporophytic phase is more elaborate as compared to the algae. Yes or no? And then this being the gametophytic phase, which is of course the dominant phase. Please tell me these points are clear to you all. Yes or no? Yes guys, please tell me, are these points clear? Yes or no? Moving on now to bryophytes. So bryophytes have two main categories. One is liverworts, second is mosses. What are the differences? See, liverworts are dorso-ventrally flattened. They are dorso-ventrally flattened and they are appressed to the substratum. What does appressed means? They are pressed towards the substratum. Is that clear? So dorsal ventral surface becomes flat, just like in flatworms. Okay. Now there are two rows of leafy shoots. I have drawn that. Two types of leafy shoots, right? So this represents the gametophyte. We are talking about the gametophyte here, right? Now, see here. The two most important examples of liverworts which are in syllabus are Markensia and Rixia. Rixia is not given, but sometimes we get a question, so just learn another name. So Markensia and Rixia. Markensia is unisexual. Now you have to learn these things. Rixia is bisexual. Bisexual means on the same thallus, both male and female sex organs will be present. While in Markensia, there is a different male thallus and a different female thallus. Is that clear? Now, the gametophyte has a leafy stage. Let's talk more about it. So this is how Markensia looks like. So it has this thalloid body, right? It has these rhizoids on the dorsal surface, and then this area, on the upper area, there is this stalk-like structure called archegoniophore. And on the top of archegoniophore will develop archegonia, that is the female sex organ. This is the female sex organ. Now similarly, on another thallus, we will have enthridiophore. 4 represents the stalk-like structure which bears something. And on the top, instead of archegonia, it will develop enthridium. Please tell me these points are clear. So the sex organs are present on different thallus. Is that clear? Okay. Now do you see there are these special structures present here which I have mentioned as gamma, and there is a cup-shaped structure called gamma cup, right? Within this gamma cup is present gammae. Now gammae are nothing but asexual buds. They are multicellular, they are green, they develop in this cup-shaped receptacle known as gamma cup. And these gamma will detach from the parent body and give rise to an independent new thallus. Is it clear? Now, since this gamma is developed, has developed on the female thallus, so this gamma will give rise to a female thallus. Had it, similarly, there will be gamma present on the male thallus also, that gamma will give rise to the male thallus because it is asexual reproduction, so clones will be formed. Please tell me these points are clear, right? Okay. Now, moving on to another class of bryophytes, which is mosses. So whether it is liverworts or mosses, the dominant parent body remains a gametophyte, okay? But the difference is, in case of liverworts, the gametophyte is very, it just have one phase, that is a leafy stage, it has one stage. But in case of mosses, the gametophyte is more elaborate. So first it has a protonema stage and then it is followed by the leafy stage. Now what are these two stages? We'll discuss one by one. Well, you should know the examples of mosses: sphagnum, funaria and polytrichum. Talking about the protonema stage. See, while I was telling you this, liverworts will have leafy stage. What does it look like? So it has these rhizoids, it has this main axis, and it has two whorls of spirally arranged leaves. This is the gametophyte, right? And it grows vertically upwards. But in case of mosses, there are two stages. First one is protonema stage. Now, protonema stage is a creeping stage. So it is going to grow horizontally on the ground, not grow vertically upwards. So it will first creep around along the ground. It is green, highly branched, and it is filamentous. It develops directly from the spore. Now see here, we have just discussed the life cycle. Do you see the spores? Spores are going to develop into parents. Now what is the parent body like? I told you, it will have a main axis, rhizoids and leafy structures, right? But in case of moss, before this leafy stage, spores are going to form a creeping stage, which will be highly branched. Is it clear? So spore will give rise to this creeping stage, and from the creeping stage, guys, will arise a bud. And this bud will then grow into a leafy stage. Are you able to understand? So the leafy stage is preceded by a creeping stage, not in liverworts, but in case of mosses. Is this clear? So protonema stage. So it develops directly from the spore and it gives rise to a lateral bud, and lateral bud will produce the leafy stage, which is upright. It has a slender axis and spirally arranged leaves. Please tell me these points are clear. So liverworts, the sex organs can be present on the same thallus. Like in case of Rixia, it is a bisexual. It can be present on different thallus, that is in Markensia, hence it is unisexual or dioecious. So these examples should be super clear. So see, I have separately drawn the life cycle of moss. Everything is same, just the difference is, now see, spore will develop into a protonema stage, right? And protonema stage will then develop into the leafy stage. So this is the first stage, this is the second stage. So parent, that is the gametophyte, which is dominant free living autotrophic haploid, has two stages. One protonema and second leafy stage. Are these points clear? Rest everything remains the same. Rest everything is the same. Please tell me these points are clear. Yes guys, please tell me these points are clear to all. Then moving on to pteridophytes, next category. Now this group of plants will have vascular tissue. The body will be well differentiated. It will have true root, stem, leaf. Is that clear? Now see, first of all, I'm going to talk about the four classes of pteridophytes. This is a very important question. The pteridophytes will be divided into Psilopsida, Lycopsida, Sphenopsida and Pteropsida. They are known by some common names. Lycopsida is known as club moss, Sphenopsida is known as horsetails, and Pteropsida is known as ferns. Now these are the examples. So almost every year we are getting one question out of this topic. One question at least. So one time it is asking the Sphenopsida belongs to, Equisetum belongs to Sphenopsida. Then there are three examples of Pteropsida. So there are multiple questions that can be formed every year. Is that clear? So please make sure that you learn these examples. It is already a PYQ. Now, so this was a question in 2025. So you should know the examples of each. So it was an easy question. So see, pteridophyte, salvinia, bryophyte, polytrichum, angiosperm, salvinia, gymnosperm, ginkgo. This was 2025 question. Now, there was another question in 2025 which was based on the correct sequence of the life cycle. So see, these were the 5 steps given and you were asked arrange them in the correct sequence. Now see, so first of all, so these are the 5 topics given. So you have to arrange in the correct sequence. So it starts with attachment of gametophyte to the substratum. This is the parent, right? So it is a gametophyte attaching to the substratum, followed by releasing of entherozoids in water, because we know gametophyte will produce gametes. This is the male gamete, entherozoids in water. What will happen? The entherozoid fusion will occur with egg. Fertilization is occurring here, followed by zygote formation, and zygote will undergo mitosis to form sporophyte, and then sporophyte will undergo reductional division to form haploid spores in the capsule region. Remember? So this was a question of 2025. So we can get similar questions now also. So make sure that you learn the life cycle in a proper sequence, and that's why I have brought these flow charts so that you form a pictorial memory also and you learn them very clearly. Please tell me these points are clear, guys. Please tell me. Yes. Now moving on to next, life cycle of pteridophytes. Now pteridophytes are the first true terrestrial plants. Is that clear? Now here it is going to shift. See, in bryophytes and algae, the parent body was haploid. Parent body was gametophytic. But now on, the parent body is going to be 2N, that is sporophytic. So parent body is going to be a sporophyte. And rest features, general features of parents, they are dominating, they are free living, they are autotrophic, they are independent, they are multicellular. And here we will have well differentiated body for the first time, true root, stem and leaf. Is that point clear? And of course the well developed vascular tissue also. Please tell me these points are clear. Quick guys, please tell me these points are clear. Yes, please. All clear here? All right. Now see, what will happen? Sporophyte. What does sporophyte do? Sporophyte bears spores. So in order to bear spores, guys, now see, the parent body, it has true root, stem and leaf. There will be some special leaves which will bear spores. Is that clear? So it is going to be very differentiated. So it has leaves meant for photosynthesis. Some leaves can be small sized, we'll call them microphylls. Some leaves can be large sized, we'll call them macrophylls. These are the examples with microphylls and macrophylls. But other than these microphylls and macrophylls, these are photosynthetic. We have some special spore bearing leaves which are called sporophylls. Is that clear? Right? Now on sporophylls, guys, are present spore bearing structures. One is a leaf, sporophyll. On the leaf, see, this is a sporophyll. On the sporophyll are present these sporangia. These are sporangia. And within the sporangia will be present spore mother cells. These are spore mother cells. And spore mother cells will produce spores. Are you able to understand the hierarchy? Yes? Now, these sporophylls can be present separately or they can club together to form one compact structure known as cone or strobilus. A very similar cone that are going to be formed in gymnosperms also. Is that clear? Now see, only in 2 cases we will see the cone formation or strobili being formed, that is in equisetum and Flagellina. Now examples are very important here. You have to learn which pteridophyte forms the cones. It is just 2, equisetum and Flagellina. Remember, Flagellina is a very special booty. It's a very special plant. It is the same plant which Hanumanji went for when, you know, Lakshman was had fallen sick and Hanumanji had to go and fly and get that Sanjeevani booty. that Sanjeevani booty was Legionella. So wherever there is some exceptional case, something special happening, Legionella will come up as an example. Remember it as a trick. Okay. So whenever there is something unusual happening in the pteridophytes, Legionella is there. Is that clear? So see, most of the pteridophytes are not going to form cones. Legionella will form that along with equisetum. The structure of equisetum is given in your NCERT. Is that clear? So let's repeat it once. The parent body with true root stem leaf, okay, they can be small or they can be large, but since it is a sporophyte and it has to produce spores, it has some special spore bearing leaves which are known as sporophyll. Phyll is for leaf. Those leaves will have these structures called sporangia. Sporangia will contain spore mother cells, again 2N. Now, meiosis will occur in spore mother cells to produce spores. And with the formation of spores, the gametophytic phase begins. So, can we say spores are the first cell of gametophytic generation, right? Now, these spores are mostly homosporous. See, what do we understand by homosporous? Means same spores. Whether it is male or it is female spore, you cannot distinguish between them. In algae, in bryophytes, and even pteridophytes. Algae and bryophytes are completely homosporous. But in pteridophytes, you will see they are mostly homosporous, but not always. We still have an exception. See, I told you, whenever there is exceptional thing happening, Legionella will be there. So Legionella and Salvinia are heterosporous. It is a PYQ already. Is that clear? Now, these spores will germinate. That means they will undergo mitosis. It is always mitosis that occurs in germination. So spores will germinate. That means undergo mitosis to form a multicellular body called gametophyte. Now, sporophyte is well differentiated, but the gametophyte will not be differentiated. Hence, we call it prothallus. But now see, the prothallus, yeah, the sporophyte in case of bryophytes was attached, right? It was attached to the gametophyte. But here, the gametophyte will not be attached, right? It will have independent existence. Now it is a gametophyte. So gametophyte will bear gametes. So that it has to have sex organs. Again, the male sex organ is called antheridium, female one is called archegonium, same terminology. And then they will form gametes, antherozoids and egg cell through mitosis because they are already haploid. Again, similar type of antheridium archegonia you can see here, jacketed. Sex organs are jacketed here. Is that clear? The two will undergo fusion. Now fertilization will occur in water. Now see, in bryophytes, there was a lot of dependence on water. Pteridophytes are first true terrestrial because the dependence on water has extremely reduced. But still, fertilization will occur in water. So, very minimal dependence on water, right? And with fertilization, zygote is formed, which is always 2N, and it represents the first cell of sporophytic generation, and it will again undergo mitosis to form the parent. Please tell me that these points are clear. Guys, let's take a quick thumbs up break here. We have discussed 3 life cycles so far. Please tell me that these points are super clear, because beyond this, after this, we are going to move on to the most advanced life cycle, that is diplontic. So please tell me these points are clear to you all. And how helpful do you think these flowcharts are going to be? Yes, guys. Will you be able to learn all things, everything in detail through these flowcharts? Yes or no? Will you be able to answer all the sequence-based questions through these flowcharts? Yes or no? Right? Right? All right. Now, moving on. So, I told you about the prothallus. Prothallus is gametophyte. It is small, inconspicuous, multicellular, free living, right? Autotrophic. Why is it free living? Because it can make its own food. It needs water for fertilization. So dependence is still there, but has been highly reduced. So it is limited to specific geographical areas. It is free living because it does not draw nourishment from the parent body. It can produce its own food. Please tell me these points are clear. Yes, guys, are these points clear to all? So you need to learn some exceptional cases. Most of the sporophylls will be separately present, but in case of Equisetum and Legionella, they will form a cone-like structure. They will form a cone-like structure. Mostly homosporous, but not in case of Legionella and Salvinia. Is that clear? Now moving on to the next, but before that, let's see the condition of spores, which I've already discussed with you. See, algae and bryophytes all are homosporous. That means male and female spores are similar. In pteridophytes, it is mostly homosporous, but exception is there, except there are some heterosporous pteridophytes, which is Legionella and Salvinia. Then, when we move on to gymnosperms and angiosperms, they will be all heterosporous. So that means male and female spores will be totally distinguishable. The male one will be small in size, so we are going to call it microspore, and the female one is going to be larger, so we are going to call it macrospore or megaspore. Is that clear? Now these microspores will also be known as pollen grains, and we are very familiar with these terms. Please tell me, will you be able to answer these questions now? So I have tried my level best, guys, so that everything written in this chapter is taken out properly and highlighted properly based on the questions coming out of this question. Are we clear? And guys, yes, you can get all these notes. You can get all these notes on my Telegram channel, which is Tina Ma'am Biology. I've already uploaded all the previous notes, also all the flow charts that we have discussed in the class. So make sure that you join this Telegram channel. Let's have a quick water break, and all of you quickly join this Telegram channel so that after the session, when I share it, you can revise the chapter from these flowcharts. Is that clear? Quickly join the Telegram channel, and we are going to move on to the next stage in the life of plants, that is gymnosperms. But now see, gymnosperms will have naked seeds. So now after pteridophytes, it is a seed habit, which is going to begin, right? Seeds will be formed. But that seed formation, that seed habit will arise itself in the pteridophytes. Now very, very important. Evolution of seed habit occurs in pteridophytes. Seed will be formed in gymnosperms, but seed habit, that means that idea that okay, in the next step now seed will be formed, that idea will be present here in the pteridophytes. See here, there are very important lines written here. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of zygotes in young embryos take place within the female gametophyte. This line is very, very important. This event is a precursor to the seed habit, which is an important step in the evolution. Now see, there are certain stages which occur during the evolution of seed. Number one, there has to be heterospory. Male and female spores should be different. So we have already seen there are megaspore and microspores being formed. Not in all, but in few at least. So it has begun. Then retention of megaspore, isn't it right? It's writing clearly. The zygote develops into young embryo, and this development occurs within the female gametophyte. So retention of this spore gradually within the female sporophyte also. Vivipary is this, right? So there are different stages, and then there will be some other things also happening that we will see in the next phase. So I hope this thing is now totally clear to you all. Sporophyte is always 2n. It will give rise to spores, and spores are n. So sporophyte produces spores through meiosis. This is just a revision so that there is no confusion regarding terms. Now spores will germinate to form gametophyte through mitosis. And if the parent body is gametophyte, it will form gametes through mitosis, and gametes will fuse to form 2n, that is a sporophyte. I hope these things are clear to you all. Now moving on to the next phase, that is the phanerogams, the spermatophyta, seed bearing plants called Gymnosperms. What are the properties? See, we call them gymnosperms because gymno means naked, naked seeds, because seeds are not enclosed within the ovary. So ovules are present, right? They are exposed. They are exposed. This was the question this year. Okay. Now, these plants, if we talk about the plant body, of course, well differentiated. It will have true root, stem and leaf. Now, these plants don't have herbs in their category. There are no herbaceous gymnosperms. They can be shrubs, they can be medium-sized trees, or they can be very tall. Okay. So shrubs are cycas. Tall trees, we have example of Ciconia. They're very tall trees. We call them giant redwood trees. Giant redwood trees are scientifically known as sequoia. And then we have medium-sized trees also. For example, pinus and cedrus. Is that clear? Now, the root system present in gymnosperms is taproot system, and they will show diarch to polyarch condition, and you know what it means. There can be 2 to more than 6 vascular bundles present, right? Then these roots show symbiotic association. The roots show symbiotic association. For example, in pinus, the roots show symbiosis with fungus, and this association is known as mycorrhiza. So, myco word is for fungus only, mycology, right? So fungus and pinus roots forms mycorrhiza. What does this fungus do? It helps in phosphorus uptake, it increases the disease resistance and also releases the growth factors. While cycas roots, the roots of cycas will show symbiotic association with cyanobacteria, that is blue-green algae, and this association is known as coralloid roots. So how do you remember? CCC. Cycas with cyanobacteria forms coralloid roots, and now here the cyanobacteria is going to play a very important role in nitrogen fixation. Please tell me these points are clear. Now again, the plant body can be unisexual or bisexual. For example, cycas is unisexual. That means we'll have a separate male plant and a separate female plant. Or it can be bisexual or monoecious, for example in pinus. So in case of pinus, commonly known as cheed, so when you go to mountains, you easily see these, roughly triangular shaped canopy. Those trees are generally pinus trees. Okay. Now, the pinus plant will have both male and female cones on the same plant body. Hence, it is bisexual, and also the cedrus. And both are medium-sized trees. Clear to all? Now, talking about the leaves. So, we said the body is well differentiated: true root, true stem, true leaf. So stems we have seen, they can grow into shrubs, medium-sized trees or tall trees, right? Also, we'll talk more about the stem. Root is always taproot system with some symbiotic association in few. Now talking about the leaves here. So leaves of gymnosperms are pinnately compound. They will have thick cuticle and it will have multi-layered epidermis with stungan stomata and needle-like leaves. Now all these are the xerophytic adaptations which are helping these plants to withstand the extreme conditions of temperature, wind and humidity. Is that clear? Now, so this is how a pinus leaf looks like. See, the pinus leaf is dimorphic. There are two types of leaves. So when we hold it close and we observe it, we will see two types of leaves. On the outer side, guys, we will have a scaly leaf. It is dry, non-green, and deciduous means it will fall off, right? But then there are some needle-like leaves coming out of the scaly leaf. These are the actual foliage leaves which are green, photosynthetic and green. So since there are two types of leaves present, we call it dimorphic. Are these clear? And it has sunken stomata. So the epidermis goes inside like this, forms this cavity, and stomata is present here in the cavity and there are bristles. This reduces the water loss. That is why I said it is a xerophytic adaptation. Now talking about the leaf scar. So on the stem, wherever a leaf is present and once a leaf falls off, a scar will always be left behind. So whatever be the case, be it cycas or pinus, let's say if a leaf is present here and it falls off, a scar will always be left behind. So you will see there are multiple marks on the stem. The stem is never very smooth. The stem is always full of marks or scars. So they are the scars from where the leaf has fallen off. Is that clear? So leaf scar is a characteristic feature of gymnosperm stem. Now the stems can be branched also, the stem can be unbranched also. It is unbranched in case of cycas and branched in case of pinus, cedrus and ginkgo. So it shows distinct secondary growth. And ginkgo biloba, guys, ginkgo biloba, this one is a living fossil. Like in animal kingdom, we have read about Limulus, the king crab. It is called the living fossil because it has never underwent any evolution. Similarly, ginkgo biloba is also a living fossils amongst the plant kingdom. Please tell me these points are clear. Yes? All right. Now moving on to the life cycle. Now gymnosperms and angiosperms both will show diplontic life cycle. That means the dominant phase will be a sporophyte. Now gametophyte, see, highly reduced, and last group of plants to have archegonia, which is flask shaped. By the way, pteridophytes are the last ones, last to have antheridia. And these are the last ones to have archegonia. Is that clear? Now learn these points. So guys, this is a table which summarizes everything. Quickly take a screenshot. This summarizes everything that we have read till now. So see the life cycle. Algae mostly haplontic, haplodiplontic, haplodiplontic, diplontic, diplontic. So here the parent means the dominant phase gametophyte. It is still gametophyte. Here it becomes sporophyte, sporophyte, sporophyte. Now the gametophyte is the one which bears gametes. So it will have sex organs. Sex organs here will be non-jacketed. We call those sex organs as antheridia and oogonia. Here the sex organs will be jacketed. Now here also sex organs wherever present will be jacketed. Is that clear? Now sex organs will be jacketed, antheridia, archegonia, and it will give rise to a sporophyte. Sporophyte will be developed but dependent on the gametophyte, multicellular and attached to draw the nourishment. It is dependent for nourishment. Talking about this, sporophyte is the main parent body. Sporophyte is a spore bearing structure. Now it will give rise to gametophyte, but gametophyte will be independent here, multicellular. Gametophyte will give rise to gametes through their sex organs. Sex organs are again jacketed like in case of bryophytes. Is that clear? And now see, I've written it. Antheridium. Pteridophyte is the last group to have antheridium. Now in gymnosperms, they are the last group. See, it is the last group to have archegonia. Antheridium is already not present here. Are we clear? Are we clear? Please tell me. So there are multiple things written in this flowchart. Quickly take a screenshot so that you can revise these things even one day before the exam. Yes, guys. These are all made by lot of hard work and effort. So I've tried to make this plant kingdom easy for you. Is that clear? Shall we move on now? You didn't get the Telegram channel. Don't worry, Peksha. Leave a comment and I'll ping there. I'll share the link in the comments. Okay? In any case, if you don't find the Telegram channel, leave a comment and I will share the link there. Now see, we are talking about gymnosperms here. So gymnosperms, as I told you, let's talk about the life cycle, right? The parent is a dominant phase, which is a sporophyte, multicellular, independent, all these things, differentiated body, right? True leaves, true stem, true root, and again it also bears a spore bearing structure called the sporophyll. It also has a vascular tissue, that is the water conducting xylem and food conducting phloem. Now see the components. Xylem will have tracheids, fibers and parenchyma, but not vessels. Vessels are the characteristic feature of the next group, that is angiosperms. Now then phloem will have sieve cells, not sieve tubes. Okay, albuminous cells, not companion cells, phloem fibers and phloem parenchyma. Now all these points will be of importance when we study anatomy. So I have brought anatomy here also. Please tell me these points are clear. Now there are again special leaves which bear spores, which are called sporophylls. And now in pteridophytes, it was just equisetum and selaginella which was forming cone. But now in case of gymnosperms, all of them, they are going to condense to form cones. So in all the plants, of all the gymnosperms, sporophylls are going to form compact structure where sporophylls will come together to form a cone-like structure called sporophyll. Yes? Now see, sporophylls will be of two types. Microsporophyll will have a separate separate sporophyll which will bear male spores and a separate leaf which will bear the female spores. So microsporophylls will club together to form male cone, they will club together to form the female cone. Are these points clear? And you can easily differentiate. See, this is a clear differentiation. How do you differentiate? So male cone also known as microstrobili or staminate cone, female cone also known as ovulate cone, right? This is smaller and short-lived, this is larger and long-lived. So these are the sporophylls. These are megasporophylls. Mega word is for female. On that sporophyll, we have megasporangium, also known as ovule. So one megasporophyll will have around 2 ovules here. Is that clear? And within this megasporangium, there is a nutritive tissue called nucellus. This is the microsporophyll, and microsporophylls club together to form male cones. And see, we have microsporangium with pollen mother cells, which will give rise to the pollen grains. Are we clear? Are we clear till here? Please tell me. Now see, here in this case, the pollination is going to occur by wind only. So it will show a nemophilous condition. And here it will be single fertilization that takes place. So this is a orthotropous, that is a straight ovule with lots of nucellus around, and this is a egg cell present, right? And this is the embryo sac. So embryo sac, just like it develops in angiosperms also. So within the embryo sac, there is an archegonia. I told you, archegonia. Gymnosperm is the last group to have archegonia. This is the archegonia within which egg cell will be present. And that's how the pollen grain comes in and it fertilizes the female gamete. Now, there are 2 male gametes here. One male gamete will fuse, other will go waste. So that is why there is single fertilization. Only syngamy will take place. So that is why, guys, there is there is single fertilization. Now, the second gamete will degenerate. Now, here what you must remember, the surrounding tissue, the surrounding haploid tissue around the archegonia has already formed endosperm. So, endosperm in case of gymnosperms is formed before fertilization. It has formed before fertilization and it is haploid. But in case of angiosperms, what do we see? Ki endosperm is a post-fertilization structure and it is triploid in nature. Now, these 2 points are very important. Endosperm here is a pre-fertilization structure and it is haploid. Please tell me these points are clear. Yes or no? Yes or no? Is that clear? So guys, this is male cone, short-lived, smaller, with microsporophyll, microsporangia, containing microspore mother cells, undergoing meiosis to form pollen grains or microspores, and microspores again forming undergoing 2 mitosis to form microspore, that is pollen grain, 3-celled microspore, which represents a male hematophyte, just like in case of angiosperms. Talking about female cone, larger, long-lived, megasporophyll, megasporangium, nucellus, 1 cell acts as a megaspore mother cell, forming 4 megaspores, and again 3 will degenerate, 1 will be functional. This is just the same. Now, 1 functional will give rise to the embryo sac, and this will be archegonia, inside which the female gamete will be present. Please tell me these points are clear to you all. And ultimately, from the embryo will be formed a seed. So, mature ovule will give rise to the seed. It will contain embryo and endosperm. Embryo is always 2N and endosperm will be haploid. Now, in case of angiosperms, we say the ovule is biteguments. There are two integuments. Here it will have single integument, just the outer one, that is testa, and it is naked. See the life cycle here. We have clubbed everything. The parent is a plant body, the parent is a sporophyte. Male cone, female cone. Male cone has these structures. Female cone has megasporophyll, megasporangium, megaspore mother cell, because 3 have degenerated. They will further undergo meiosis, right? Undergo meiosis to form microspores and megaspores. These 1 functional will form the embryo sac, right? Will have archegonia, egg cell, and then this microspore will form the pollen grain and wind pollinated to undergo fertilization to form zygote, embryo, seed, and so on. Please tell me that these cycles are clear to you all. Please tell me these points are clear to you all. These cycles are clear to you. Afan Shah, how can you swear? Huh? Is that clear? Is that clear? Quickly let me know. I hope this is clear. All right. Now, all of you who are listening to me clearly and not wasting time in the chat, I want all of you to please do me a favor. Let me know in the chat once and for the final time, is the voice absolutely normal? I want the serious candidates who are listening to me quietly and writing down their notes, understanding the things. I want that crowd to answer me in the chat. Is the voice absolutely normal? I don't want the students who are wasting their time to chat here. I want the serious candidates who are going to clear NEET, Re-NEET with better marks. I want them to answer. Yes, very clear. It's perfect. Okay. So from now on, I am not going to pay attention to the useless chat here who are just spamming and wasting my time and taking away my focus. So see, I look at the chat so that your questions don't get unnoticed and unanswered. So don't uselessly keep spamming. It is clear? Thank you so much. All right. So now finally we have the most developed plants, which are angiosperms or the flowering plants. And these flowering plants, guys, these flowering plants will produce seeds which are enclosed within the fruit. We'll talk more about angiosperms in the other chapters. So parent body is sporophyte dominant, this. Now these will bear flowers. So till gymnosperms, we see there are sporophylls, right? Now instead of sporophylls, they will bear flowers, because basically flowers will also have sporophylls. Flowers are modified leaves, right? The floral parts are basically modified leaves. So stamen is a sporophyll, pistil is a sporophyll. So see, flowers will have microsporophyll known as stamen, and it will have megasporophyll known as pistil. These are the structures, and we'll be discussing more about it in the chapter of sexual reproduction in flowering plants. Let's have a quick look on the life cycle. So this is a parent, right? So parent will bear the flowers, and flowers contain the sporophylls. See here. Now it will have megasporophyll, it will have microsporophyll. In megasporophyll pistil, we have ovary. Ovary contains ovules. And what are the ovules like in case of angiosperms? Anatropous, that is inverted. Bitegmic means two integuments. Megasporangium, ovule is also known as megasporangium. It contains nucellus, which is 2n. One cell will act as a megaspore mother cell. 4 megaspores are formed by meiosis here, right? 3 will degenerate, 1 remains a functional. This is the in short story of sexual reproduction in angiosperms, right? So 3 mitosis to form 7-celled 8-nucleated embryo sac. This is a typical of angiosperms. We have just one functional female gamete, right? Which is surrounded by synergids. This is how the embryo sac looks like. So this was the story of female development. Now coming on to microsporophyll, it has anther. Anther contains pollen sacs or microsporangium, which contain microspore mother cells. Again, they will undergo meiosis to form 4 microspores, all of which will be functional. They will undergo mitosis to form microspore, and they will contain 2 male gametes. Now here there are 2 male gametes, we have 1 female gamete. So there will be double fertilization. Now this is a unique feature. right? So all these star marked things are the unique features which are occurring differently in angiosperms as compared to the gymnosperms, right? So double fertilization. One male gamete will fuse with the female gamete to form zygote, while another male gamete will fuse with the central cell to form pen, right? Here male and female gamete will fuse, and here male gamete will fuse with the central cell to form diploid structure called angiosperm, and here a triploid structure called angiosperm, and here diploid structure called embryo. So embryo and endosperm is going to feed or provide nourishment, and they will form a seed, and seed will again give rise to a new parent plant called sporophyte. Please tell me these points are clear. Is this point clear? All right. Moving on now. Moving on. So the smallest angiosperm is Wolffia. It is so small, it is a dot-like green structure. It is that small. And the tallest one is Eucalyptus. Are we clear? So guys, this is a table to differentiate between gymnosperms and angiosperms. Take a screenshot and please make sure that you revise this. This is for your revision, and make sure you revise it. I hope you've taken the screenshot, or otherwise you will of course get the notes on the Telegram channel. These are the types of seeds, and now guys we are going to move on to the next chapter, which is biological classification. Yes guys, we're going to move on to the next chapter, biological classification. Quick. Are we all clear till here? Let's have a thumbs up break. So 2 hours we have given to plant kingdom in its revision. Imagine when we do it for the first time, it takes so many hours. So I have basically used the exactly same notes which I use in my detailed lectures, right? So we are discussing everything in detail from 0 to hero level. That's what I say, right? So I'm not missing any point, and these flowcharts will help you revise NCERT in a very easy manner. Clear? All right guys. All right, moving on to the next, that is biological classification. Let's first talk about the systems of classification. See, this is something we have already discussed. So every time I start with classification, I talk about these systems. I've discussed already artificial system given by Linnaeus, right? This was superficial. This was based on superficial morphological characters. Hence, it was less authentic. Already discussed. What were the superficial features like habits, habitat, right? Then natural system given by Bentham and Hooker. This is of course more authentic because it is not only considering the external features but the internal features and phylogeny. So these features are more static, right? So it considers genetics also. So hence more stable bases are considered here. Phylogenetic, we've known evolutionary history, numerical taxonomy, 100 plus characters plus computers are utilized. Cytological cell structure, chromosome number and behavior. Chemotaxonomy is based on chemical composition. Is that clear? So this is what we have discussed already. Okay, coming on to now different classifications. So early classification, I'm not talking about the scientific classification. Human is bound to classify things. This is how so this is our disposition. We will classify things. Now the early classification was based on the use of organisms, whether this is used for housing material, or that means a shelter, or it is used for food, or it is used for clothing. So this is how we used to classify the things. But the first scientific classification was given by Aristotle. Aristotle also differentiated the organisms into two categories, two kingdoms, that is plants and animals. But the basis was, for plants, it was simple morphological features, right? And he said the plants will be herbs, shrubs and trees. And for animals, he used RBC as a criteria. The animals with RBCs and without RBCs, we call them enema and anema. So anyways, so that was a very Simple basis. But the first scientific classification was given by Aristotle. Now, although I have written he gave two kingdoms, but whenever they ask you who gave the two kingdom classification, answer should be Linnaeus, because the proper classification of organisms into two main kingdoms, Plantae and Animalia, was made by Linnaeus. So what was the criteria? The criteria was the cell wall. If the cell wall is present, plants. If the cell wall is absent, it will be animals. Is that clear? Now, according to Linnaeus, and on this basis, on this criteria, Linnaeus had put bacteria, blue-green algae, fungi, gymnosperms, bryophytes, pteridophytes, angiosperms, all under one kingdom, that is plants. So according to Linnaeus, fungus is also a plant, and algae, unicellular or multicellular, is also a plant. Clear? Bacteria is also a plant. But he kept protozoans, that means the unicellular organisms, amoeba, plasmodium, all of them, he kept all of them under animals. So animals had there were two varieties of animals, unicellular, which were known as protozoans, and metazoans, that means multicellular animals. So they were without cell wall. So all without cell wall are called animals. Are these points clear? Now, of course, there were lots of disadvantages. There were lots of loopholes. Why? You see, according to two kingdom classification, bacteria, which is prokaryotic, has been kept along with the eukaryotes. So there is no no differentiation between prokaryotes and eukaryotes. Then bacteria is unicellular. This is unicellular, or this can be multicellular, while others are multicellular. This is unicellular, this one is multicellular. So there is no differentiation between unicellular and multicellular also. Also, he kept fungus, which is heterotrophic, along with some autotrophic. So what is the criticism here, or what is the disadvantage of two kingdom classification? There is no differentiation between number one, unicellular and multicellular organisms. They have been kept together. No differentiation between prokaryotic and eukaryotic organisms, and no differentiation between autotrophic and heterotrophic organisms. Please tell me these points are clear. Yes or no? Yes? No differentiation between these. Then came three kingdom classification. Now, what was the advantage? What happened during three kingdom classification given by Haeckel? He separated eukaryotic unicellular organisms from rest of them. So see, no matter how many kingdoms do we put classify the organisms into, but you will always see that plants and animals are two constant kingdoms. Okay. So what did Haeckel do? Haeckel took out unicellular eukaryotes from the plants. See here. So there were unicellular eukaryote plants here, algae. So he took out this one, and he took out these protozoans, unicellular eukaryotes from animals, and together put them in third kingdom, that is Protista. So from animals, he is bringing the protozoans, and earlier protozoans were in kingdom Animalia, but now according to three kingdom classification, protozoans will be brought under Protista. So this is a question, where are protozoans earlier? According to two kingdom classification, animals. According to three, Protista. And then from plants, he took out unicellular algae. Earlier kept in plants, but now classified under Protista. Is that clear? So for example, Paramecium, amoeba, chlorella, spirulina, chlamydomonas. So you get these questions. Please tell me these points are clear. You get these questions, right? Okay. Now, then came still, see, even though we have separated unicellular eukaryotes, but still fungus is still under plants. Bacteria is still under plants. Then came Copeland. What is he going to do? This Copeland is going to separate the prokaryotes. So he is going to define a separate kingdom for prokaryotes. So they are unicellular and prokaryotes and form this kingdom Monera. Is that clear? That is the 4-kingdom classification. Then we got 5-kingdom classification. In 5-kingdom classification, guys, we have uh, according to R.H. Whittaker, we have 5-kingdom classification where he separated fungi. So the kingdom which was invented by Whittaker was actually fungi. So he separated fungi also. That why not fungi? Fungi is not autotrophic, right? So now then came the 6-kingdom classification. See, in 6-kingdom classification, we are going to differentiate Monera into 2 sub-kingdoms. Okay, so basically the 5 kingdoms, Monera, Protista, fungi, plant and Animalia, according to Whittaker. So according to the 6th kingdom, this Monera will be divided into Archaebacteria and Eubacteria. So this is kingdom 1, this is kingdom 2, this is kingdom 3, Protista, this is kingdom 4, this is kingdom 5 and this is kingdom 6. Is that clear? So according to 6th kingdom classification, which kingdom was divided further? It was Monera, Archaebacteria, Eubacteria. Is that clear? Now, this 6th kingdom can also be known as 3-domain. So 6th kingdom or 3-domain system or 3 domains, right? What are the 3 domains like? So Archaebacteria is called domain 1, Eubacteria is given domain 2 and rest all the eukaryotes are given domain 3. So one domain belongs to eukaryotes known as eukaryota, while the 2 domains belongs to prokaryotes. These are all questions. How many domains belong to prokaryotes and how many belong to the eukaryotes? So answer should be very, very clear. Is that clear? Now moving on to Monera. Wait. Where is Monera? Okay. So guys, I am skipping Monera from here because I am going to discuss the structure and the details of Monera along with the cell. Is that clear? So let's come directly to Protista here. So Protista is going to include unicellular, eukaryotic, photosynthetic as well as non-photosynthetic, autotrophic as well as heterotrophic organisms. Now we have further divided Protista into 5 categories: chrysophytes, dinoflagellates, euglenoids, protozoans and slime molds. Now listen, this one flowchart is going to cover everything. Listen. Now this kingdom Protista does not have very sharp boundaries. This is one kingdom. This is a kingdom which brings together all other kingdoms. One kingdom that brings all other kingdoms together. All kingdoms together. How? See, chrysophytes, they are plant-like. Dinoflagellates will be plant-like. Euglenoids is a connecting link between plant and animal. So they will be plant-like as well as animal-like. Protozoans are strictly animal-like and slime molds are both plant-like and fungi-like. So do we see that there are no clear boundaries and they will show some features resembling the other kingdoms as well. Now, what are those features? Let's discuss. Starting with chrysophytes. Chrysophytes are photosynthetic. They are autotrophic, right? They will have cell wall, and all these features are plant-like features. Photosynthetic, autotrophic, cell walls, right? But the difference is why we have not kept it under kingdom Plantae, because the cell wall is not made up of cellulose. The cell wall contains silica, which makes the cell wall indestructible. Now, the members of chrysophytes are, guys, diatoms and desmids. Desmids are also known as golden algae, and it is in the cell wall of diatoms that silica is present. Now, when the organism dies, the outer skeleton, the outer cell wall still remains. So that is why the soil which contains lots of diatoms, after the death of organisms, after many years, that soil becomes gritty because of silica, and we call such soil as diatomaceous earth or Kieselguhr. Is that clear? And these chrysophytes are the chief producers of oxygen, right? What is the function of diatomaceous earth? They are involved in filtration, polishing, and sound, forming the soundproof material. Is that clear? Then moving on to another type of protista, which is dinoflagellates. Again, they are photosynthetic. Again, they are autotrophic. This is plant-like feature. They also have cell wall, which is made up of cellulosic plates. Now, there are two very important points to be noticed here. Now, see. Remember, the cell wall of silica, these silica, they are going to form soap box like structure. They are going to fit into each other. The two plates are going to form or fit into each other like soap box. The cell wall plates, there are two only, which are going to fit into each other like a soap box. You will not hear the soap box like feature anywhere else. So the moment you think about or you read this word soap box like, this term chrysophytes should click in. Is that clear? All right. When we talk about the cell wall, cellulosic plates of dinoflagellates, so there are many plates, and the body is made up of many cellulosic plates like this. Is that clear? So now see, this is how the body is present. This is a general structure of dinoflagellate. Now, why do we call it dinoflagellate? Because there are two flagella. So the name comes from two flagella. One flagella is transverse, so it runs like this from the groove, and then the other flagella is longitudinal. Is that clear? So this longitudinal flagella and this one is the transverse flagella. It runs through the transverse furrow or the groove. Is that clear? Now, dinoflagellates are mostly marine and they have different types of pigments like red, blue, green, brown and yellow. Is that clear? Now, we have a very important example of dinoflagellate, that is Goniolux. Goniolux, given in your NCERT. Now, what is so particular about Goniolux? Goniolux is red in color. Goniolux is red in color. So the water which is bringing lots of Goniolux, it appears like a red velvet or red tide. It's like the tide is bringing red velvet around towards you. Now, whatever looks beautiful may not always be beautiful or safe, right? So Goniolux, this causes red tides, and the fishes which intake this water containing Goniolux, they die. It is toxic to the fishes. Toxic to the fishes. These are the 2 points that you must remember about Goniolux. Is that clear? Now, talking about the third category, which is euglenoids. Now, euglenoids, I told you, they are animal and plant-like. Animal and plant-like. So, basically, they act like a connecting link between plants and animals. How? See, when the light is present, they will become autotrophic. They'll make their own food, which is plant-like feature. But when there is no sunlight, it will be heterotrophic and it will intake the organism. It will show predation, right? Which is animal-like feature. So this is plant-like feature. This is animal-like feature. Is this point clear? Another, it does not have a cell wall. Again, animal-like feature. Now, instead of cell wall, guys, it has a protein covering which is flexible, and this protein covering is known as pellicle. It is a flexible covering. Now, mostly found in fresh water, but not the running water. It is found in the stagnant water. There are two flagella again. Unlike this, which is transverse and longitudinal, it has two flagella. One is short, another is long. Is that clear? And then the pigments are identical to the higher plants. So for photosynthesis, it has exactly same pigments. Is it clear? Now, moving on to the third category, fourth category, we have protozoans. So, protozoans were earlier kept in kingdom Animalia. They were kept separately in Protista. Now, protozoans, they are animal-like, they are heterotrophic. Now, that is the animal-like behavior. How does it take in food? It is holozoic. It takes the whole food and digestion occurs within the body. It is saprotrophic also, and some of them are parasitic. Cell wall is completely absent. They are called primitive animals. Now, these protozoans are further differentiated on the basis of locomotory structures. On the basis of locomotory structures. Is that clear? So those which have pseudopodia, they are called amoeboid. Then there are others which use cilia, so we call them ciliated protozoans. Then those which use flagella for movement are called flagellated. And then there is the fourth type which don't have any specific locomotory structures, but they will have a specific stage in their life cycle which is called spore-like stage. So we call them sporozoans. Is that clear? So the name itself says they will have pseudopodia. What are the organisms? Amoeba, entamoeba, right? Entamoeba is parasitic, it causes disease, while amoeba is free living. Then ciliated, in ciliated we have paramecium, the slipper organism, the whole body is surrounded by many cilia which helps in swimming. Then flagellated, the example given in your book is trypanosoma, which is again parasitic and causes sleeping sickness. And in sporozoans, we have plasmodium, the malaria causing organism, right? Then talking about slime molds. See here, slime molds are fungi-like and plant-like. They are heterotrophic, they are saprophytic, just like fungi. Now, what is the plant-like feature here? They have true cell wall in their spores. Now, presence of spores is a fungus-like feature, but having true cell wall is a plant-like feature, right? And now talking about the spores will be formed in the fruiting body. Spores are dispersed by air currents. They are dispersed by air currents. They are very strong. They are very resistant. So they are not going to get destroyed. They are very resistant. Please tell me these points are clear to you all. Also guys, talking more about slime molds, during the favorable conditions, they are going to form an aggregation. So, just like when the favorable conditions are going on, we also like to get together and sit and chit chat. So similarly, these slime molds during favorable conditions, they will get together. Their aggregation is known as plasmodium. Now this plasmodium is different from the plasmodium that I have discussed under protozoans. The difference is the spellings are the same, but if you started with small p, it will represent the plasmodium of fungi, while if it is the name of organism, it has to be written in italics or underlined with starting with a capital P. But see, as soon as the unfavorable conditions appear, plasmodium, see, this is plasmodium, they are aggregated together, right? And this plasmodium can spread to many feet. They will not have cell wall and it is multinucleated. But as soon as the unfavorable conditions begin, this is going to give rise to some fruiting bodies. So plasmodium will give rise to the fruiting bodies inside which spores will be formed. This is a fruiting body in which the spores will be formed. And spores, as I told you, true cell wall dispersed by air currents, and they are very, very resistant. Are these points clear to everyone? Please tell me, guys. So these are the protozoans that we have talked. And now coming on to the third kingdom, which is kingdom fungi. Now see, all are heterotrophic. They can be saprotrophic and act like decomposers. They can be parasitic and cause diseases. They can be symbiotic and can be nice, like lichens and mycorrhiza. Now those which are parasitic, we have white spots on mustard caused by albugo, Puccinia causes rust, and Eustilago causes smut. Is that clear? Then fungi, mostly multicellular, but few are unicellular like yeast. This is an exceptional case. Mostly multicellular. Now those which are multicellular will have a thread-like body, that means filamentous, and their filaments are known as, threads are known as hyphae. Is that clear? Now the network of hyphae are going to form mycelium. Is it clear? So one thread is a hypha and their network is a mycelium. Is that clear? This is a mycelium. So this is a hypha and the network will form mycelium. Are these points clear? Alright. Now the mycelium can be further of many types. It can be branched or unbranched like this. This is unbranched. This is having branching. This is showing branching. while it can be septate or aseptate. Septate means, see, cell wall has to be present. Cell wall is present around the whole filament. But within the structure, if the cross walls are present, now these are the cross walls, then we call it septate. If the cross walls are not present, we call it aseptate. Now, if the cross walls are not present, this is aseptate. So what will happen? All the nuclei are present in the continuous, all the nuclei are flowing in the continuous cytoplasm. So aseptate condition will always give rise to the multinucleated condition, which is known as cenocyte. Please tell me these points are clear. Very basic, very basic. Right? Now, talking about reproduction in fungi, let's talk more. So reproduction in fungi can be of three types: asexual, vegetative, and sexual. Now, vegetative is by three different ways: fragmentation. So filaments will break and every filament, every hypha will give rise to the new mycelium. Then budding, which occurs in case of yeast, and then fission also. Now, talking about asexual reproduction. In asexual reproduction, spores will be formed. Now, these spores are different from the spores formed during sexual reproduction. So, learn the names very carefully. So, guys, in see, what are the asexual spores? So, these are asexual spores. The different names of asexual spores are conidia, zoospores, sporangiospores. Is that clear? Now, see, we have 4 classes in fungi. We are going to remember it as PAD-B: Phycomycetes, Ascomycetes, Deuteromycetes, and Basidiomycetes. Now, Ascomycetes will have, Phycomycetes will have what type of asexual spores? Let me know in the chat. What type of asexual spores are present in Phycomycetes? Please let me know. But this A represents that they will have asexual spores present, asexual spores present. But Basidiomycetes do not show asexual reproduction, hence it is absent here. Yes, guys. Quickly let me know what is the name of asexual spores in Phycomycetes. Quickly. So it has zoospores. Yes. While this is going to form conidia and this is also going to form conidia. Now look at this carefully. So see, conidia. So if this is a mycelium, so how does conidia develop? So there is a vertically upward growing structure, right? And on the top of it is this swelling, which is going to become a conidia. And again, another swelling. So these are all conidia. So they are formed on the outer region. Hence, conidia are always exogenous. Please understand, they are always exogenous. But if I talk about zoospores, then zoospores are formed in a sac-like structure like this. So this one is known as sporangium, and inside which the spores are formed, called the zoospores. Please tell me these points are clear. Everyone. Please tell me these points are clear. Right? So zoospores are formed within the sac-like structure, hence zoospores are endogenous. They are called endogenous. They are formed inside. And in Basidiomycetes, there are no asexual spores. Now talking about the sexual spores, just remember them by the name of boa. Boa is ajgar, a snake. So boa stands for Basidiomycetes, oospores and ascospores. Is that clear? Now, see, this is boa. So for Phycomycetes, PAD-B represents Phycomycetes, Ascomycetes, Deuteromycetes and Basidiomycetes. Now see, all of them will have sexual reproduction except Deuteromycetes. So Basidiomycetes will not show asexual reproduction, while Deuteromycetes will not show sexual reproduction. But yes, it is not rightly said. Sexual reproduction has been found later on in Deuteromycetes, but that stage was so dissimilar from the parent Deuteromycetes that they were kept under a different classification, mostly under Asco and Basidio. Is this point clear? Right? Now there are 3 steps. In which sexual reproduction is going to take place. Very important. 3 steps. Number 1 step is plasmogamy. So basically the two hyphae are going to fuse. Just the cytoplasm is going to fuse. This is known as plasmogamy. The nucleus will not fuse, right? So when just 2 hyphae will fuse and not their nucleus, it will form a dikaryon phase, that is N plus N. Is it clear? Later on, the nucleus will fuse. So see, generally what happens, first the cytoplasm fuses and automatically and simultaneously the nucleus also starts fusing, but not in case of ascomycetes and basidiomycetes. So these steps are being followed by these 2 classes of fungi. So first of all, just the cytoplasm will fuse, the nucleus will not fuse immediately. So this results in dikaryon stage. That is why we write N plus N. So 2 nuclei looking towards each other, not fusing. Dikaryon phase, right? Gradually, now the nuclei of the 2 hyphae, they decide to fuse. When the nuclei fuse, this is known as karyogamy, and this is what will cause zygote formation. And finally, zygote will undergo meiosis to give rise to spores. So this is already a PYQ. So if you are asked, arrange the following stages in the correct sequence, so you should know first comes plasmogamy, then dikaryon, followed by karyogamy, giving rise to zygote, zygote undergoing zygotic meiosis and forming the spores. Please tell me these points are clear to all. Is that clear? Is that clear? Alright, so this is how I have shown the plasmogamy. See, this is one mycelium, this is another mycelium, this is plasmogamy going on. So the plasmogamy and so it will give rise to a secondary mycelium where it will show mitosis and form a new mycelium. But do you see the nuclei of the 2 different hyphae or the 2 different mycelium have not fused yet, right? So this is the dikaryon stage. This is how a dikaryon stage looks like, right? And then after a while, the 2 nuclei will fuse to give rise to zygotes. So these are all zygotes. Yes, this is karyogamy and giving rise to zygotes. And then finally, zygotes will undergo meiosis to give rise to spores. Now remember, in ascomycetes, guys, in ascomycetes, so we know one cell will undergo meiosis to form 4 cells, right? So technically there should be 4 spores formed, but not here. Why? Because in ascomycetes, the 4 spores formed will again undergo mitosis to give rise to 8 spores. So 8 spore formation is a typical feature of ascomycetes. Is this point clear? So this is in case of ascomycetes, not basidio. This is in case of ascomycetes. Please tell me these points are clear to you all. So guys, this is the table which summarizes everything given in fungi. So first the examples, how do you learn it? Pram. P for Phycomycetes, Rhizopus, Albugo, Mucor. Rhizopus is the bread mold, Albugo causes white smart spots on mustard, right? There is no mnemonic for ascomycetes. If you can make, you can share with me in the comment section, but I don't require, and I feel if you learn other examples, rest of all the examples left over are kept under ascomycetes, okay? So we have DCAT to learn the examples of deuteromycetes and BAPU. BAPU means father. So BAPU for basidiomycetes. Is that clear? All right. So where are they present? What type of hypha are present? So only in Phycomycetes we have aseptate and cenocytic condition, while in rest of them it is septate and branched hyphae. Is that clear? Told you in Phycomycetes we have motile spores known as zoospores and aplanospores as the non-motile spores, and both of them develop within the sporangium. So they develop endogenously, while in this and this we have conidia formation. They will not show asexual spores. So here and here it will be conidia. So in both the cases conidia will be formed on the outer side, hence exogenous. Is this clear? And then, so this is ascomycetes already discussed. And then last not the least, see, animal kingdom is discussed in detail, plant is discussed today itself. Now last not the least, we come on to virus, viroids and prions. See, virus, the body contains protein coat known as capsid and it has the genetic material. It can be RNA or DNA. Both RNA and DNA are never simultaneously present. Now see, there are different types of genetic material. So the genetic material can be double-stranded RNA, double-stranded DNA and single-stranded RNA. So which type of genetic material is found in bacteriophages or plant viruses and animal viruses? You should be very, very thorough with this. You should be very, very thorough with this. Is that clear? Now, this is your homework. You have to answer me in the chat or in the comment section that which type of genetic material is found in bacteriophages while in plant viruses and animal viruses. Now then, there are few viruses which will have an additional covering known as envelope. It may or may not be present. If present, it is made up of glycoproteins. It is known as, if present, it is made up of glycoproteins. Is that clear? Okay. Now, talking about viroids. Viroids, unlike virus, which has protein, capsid and genetic material, viroids will not have any capsid, no cover. So it is free RNA. It is naked. And because there is no capsid, it will automatically have a smaller size. It is smaller than the virus and it will have low molecular weight. Is that clear? Okay. Now the very important example of viroids is that it is also disease causing and it causes a disease known as tobacco spindle tuber disease. So this is the least you have to remember about viroids and they were discovered by T.O. Dinier. I'll talk about the scientists in a while. And talking about prions are, viroids don't have capsid, prions don't have genetic material, they will have abnormally folded proteins and they are disease causing, pathogenic, and most of them are known to cause the neurological disorders. They are known to cause neurological disorders or diseases. Like in cows, it causes mad cow disease. Now the prion, which causes mad cow disease, we call it bovine. This disease is also known as bovine spongiform encephalopathy. Is it clear? Encephalon is the word for brain. So bovine word is for cattle, cows. Now see, the variant of mad cow disease causes a disease in humans which is known as Creutzfeldt-Jacobs disease. It is the CR stands for Creutzfeldt. It is given in new NCERT, Creutzfeldt-Jacobs disease. Are these points clear? Are these points clear? So quickly let me know guys if these points are clear and yes. Are these all points clear to everyone? So I am going to discuss all the scientists separately guys. I have already made a video before May 3rd for your NEET exam in which I have discussed the scientists in the form of flashcards. So I've tried to make it very easy for you to revise. So go back in the list and try to revise all the scientists. Also, I will be revising that I will be taking a separate session on scientists once again because we are bouncing back, we are revising everything for Re-NEET. But if you want to quickly revise through flashcards, that video is already there. Is that clear? But we will do that again also in the upcoming videos. So guys, we have completed biological classification. We have completed plant kingdom as well. Is that clear? So guys, now it's time for you to understand that there are students who are going to appear for NEET 2027 and maybe some of you have also decided to reappear in NEET 2027, right? Take a drop. Of course, you will take chance in NEET 2026, but still there may be few who are very sure that, okay, We'll take Re-NEET, but we will again take exam in 2027. For all those students, we are starting a Phoenix 2.0 batch, and you can get that for only less than 4,000 rupees if you use the code TRLive. Is that clear? And what all are you going to get in these batches, guys? The batch is not going to start on 30th May now. This batch will be started after your Re-NEET exam. So yes, it has been delayed because of your Re-NEET. We don't want you to get distracted, but the batch will be starting after the Re-NEET exam. This will be for class 12 and dropper. There are many English batches, just one English batch, which will be taken by me and my team. Yeah, and you can enroll in this batch by using that code and get it only for less than 4,000 rupees. Is that clear? Is it clear? And what do you get in all that? You get regular doubt solving sessions. So after every 2 to 3 lectures, we have a separate doubt solving session. Access to Prime, and you get all the ongoing and upcoming batches. It's not that if you take a batch, then you can only attend my classes. You can attend other educators' classes also. You get digital notes and practice modules, all the test series, practice questions, more than 15,000 questions, and you get all the PYQs as well. So make sure that you share this information, and you also remember this information for yourself if you are planning for NEET 2027. Now, I think we should stop, and we will take back from here in the next session. The next session will be day after tomorrow at 7 p.m. Is that clear? Not tomorrow, because tomorrow I am going to take a plus class at this time. So tomorrow, day after tomorrow, I am going to take the third part of botany of class 11, and we are going to complete our morpho, anatomy, and cell biology. Is that clear? So we'll take those 4 small chapters in the next chapter. In the next session, and I'll see you then. So make sure that you learn all these topics properly. And tomorrow, guys, at 10:30, I am also going to take a special class where we will be practicing these questions. Clear? So see you in the special classes, and see you in the part 3 of botany day after tomorrow at 7 p.m. Thank you so much, and all the very best, and keep studying.